Good afternoon. Today's lecture will be the first one in the series of 3 lectures which will deal with stereochemistry. Stereochemistry is a branch of chemistry which studies three-dimensional structures of molecules and implications that that three-dimensional structure has on reactivity of molecules. And as such, this branch of chemistry has immense applications in biology, physiology, material science, and pretty much any branch of modern chemistry today. And a good point to, and we will now talk about stereochemistry today and probably next week as well, but we will also talk about stereochemistry each time we start talking about new class of compounds, and we'll revisit the stereochemical intricacies of that specific class of compounds. Now a good point to start discussing stereochemistry probably the concept of isomers. I mean, we already introduced isomers as compounds which have same constitutions, or same number of carbons, hydrogens, oxygens, and other elements, but different structures. We also introduced 2 subgroups of isomers, one being constitutional, which have different bonding sequence, and an example would be molecule of cyclohexane versus molecule of hexene, both of which have the molecular formula C6H12, but they have different structures. And the other type of isomers were stereoisomers. And these had the same bonding sequence, but different orientation in space of different parts of molecules. And we also introduced 2 examples, one being the trans-2-butene and the other one being the cis-2-butene, where all the bonding sequence is different, it's just that the orientation of these groups in space is different. So you might think, well, these 2 are different compounds, that's all very nice, but how big is this difference anyway? And a good place to illustrate with this would be a comparison between structures of these 2 carboxylic acids. One of them is trans, and this is the so-called fumaric acid. Its melting point is actually quite high, 287 degrees centigrade, and it's an essential metabolite that occurs in our bodies. Its cis isomer is the so-called maleic acid. Because of its cis configuration, its melting point is significantly lower since it packs much worse in a solid state, and this is a toxic and irritating compound. So certainly not an essential metabolite. And so you can see that for these 2 compounds, the structure determines physical properties, but also biological interaction. And so this difference is certainly significant. And so stereoisomer come in many flavors. One that we already experienced was cis-trans. But today we will talk about mirror image isomers, which sort of introduces to a much larger concept in chemistry known as chirality. And our next topic will explain what chirality is.